Hi, everyone. Hope everyone's having a good weekend. This is Caroline with Fiveable. So tonight, if you have been following along, we are talking about tonicity and osmoregulation, okay? And if you don't know what those words mean, you actually probably do. We're really just going to be talking about water balance and the movement of water, osmosis, things like that with concentration gradients. And I do have some practice questions for you as well. And I'll be sharing my screen with you and doing some written things as well. And then in this particular session, we'll do a review of the plasma membrane. We'll talk a bit about concentration gradients. We'll talk about tonicity and osmoregulation, and I'll give you some real life examples of that as well, because that always makes things more interesting. Okay, so just briefly, and if you tuned into my session, I think it was last week, a week ago, then these are all review for you. But just a quick review, we can't talk about osmosis if you're not super familiar with the plasma membrane. So there's our plasma membrane. It has a lot of components to it, but most importantly, it has phospholipids, okay? So that's what we're gonna be talking about mainly. And then those proteins that are stuck in there, we're gonna use those for osmoregulation as well. So the phospholipid, the important thing that you need to know about it is that the phosphate group, which forms the head, which is the red dot on your screen, those are hydrophilic, meaning water loving. They are polar because a phosphate group has an unequal distribution of charge, and that makes them water loving. And then those fatty acid tails, which are the yellow squigglies that you see in the inside, those guys are hydrophobic or water fearing, and they are going to regulate what can and cannot get through the cellular membrane. So water is polar. So if water is trying to get through and all of these tails are water fearing, right, it's going to have a hard time getting through the plasma membrane on its own. In fact, it doesn't really get through on its own except for like a molecule here and there. So we're going to need to use a transport protein to get our water in and out of our cell, okay? And again, that's because of the hydrophobic nonpolar fatty acid tails that exist in the inside there and control what comes in and out. Okay. Oops. All right, and then just as another quick review of our 4 different types of transport. So simple diffusion is passive, meaning that we do not use energy, and that's for small nonpolar molecules that are moving with their concentration gradient. Facilitated diffusion is what we will be talking about today because we are talking about small polar molecules. We're talking about water. It's also passive, meaning no energy is required, and we're moving with the concentration gradient. And then active transport would be if we're moving against the concentration gradient or moving something that's large. And then endo and exocytosis are things coming into and out of the cell that are large and bulky, and that will also need energy. So for tonight, when we're talking about facilitated diffusion, you need to be familiar with a concentration gradient. So we'll talk about that in a second. But any questions so far? Like I know I just went through that very, very quickly, only because we've talked about it so much over the past like 6 sessions. We all okay to jump in? Okay. So facilitated diffusion, slightly more up close. You will notice that our concentration gradient, we have lots more green particles, whatever they are, on top of that cell as opposed to inside of that cell. So the extracellular fluid has a higher concentration than the intracellular fluid, which is why using that fancy dancy channel protein, because remember this is for polar molecules that can't get through the membrane on their own, they are moving inside of the cell, okay? Things will always move from a high concentration to a low concentration. And I always give the mountain analogy. If you're coming down the mountain, no energy is required. If you're going up the mountain from low concentration to high concentration, that takes a lot of energy, okay? So this is the kind of process that we are discussing specifically with the movement of water. So water has their own channels called aquaporins. So I'll write that into our chat box. Aquaporins, okay, should make sense. Aqua, water, porin, pore. So this is a pore that water can get through, and you'll notice that it's actually positively charged on the inside, and that kind of pushes the water through. It's attracted to the positive charge, and it's pulled through that electronegative oxygen is very happy to travel through these channels from a high concentration to a low concentration. So what does this all look like? So tonicity, right, just has to do with concentration gradients and therefore how water moves in response. So you've probably heard these terms before, even if you didn't know it was called tonicity and osmoregulation, but you've heard of a hyper, iso, and hypotonic environment before. Soleil, that's a great question. Are aquaporins in all cells? Yes, they are, because all cells, no matter what, need to move water. Doesn't mean that water is always going to move in the same direction, but all cells move water. Okay, we need water. It's a byproduct of cellular respiration, so it's something that we make from cellular respiration. So if we couldn't get rid of it, our cell would burst. And we also need it for lots of things in our metabolism. So yes, all cells have aquaporins. Great question. Okay, so our three different scenarios here, hyper, iso, and hypotonic, all have to do with concentrations, okay? So we're gonna talk about the concentration of water, and we're gonna talk about the concentration of solute. So when I say solute, if you're not familiar, it's really just anything that's dissolved in water. More often than not, we think of things like salt and different ions, potassium, sodium, those are the big ones. Just because potassium and sodium and calcium and magnesium, those are the biggest ions that are in our body. And their concentration is going to depend on the way that water moves. And water is going to depend on the concentration of all of our extracellular ions. And I can give you some examples that might make a little bit more sense of that. Like when you're super dehydrated, if you have really bad diarrhea, which we don't like to think about, but it happens sometimes, then these concentration gradients can shift. So before I go in and explain all of these things, I just want to quickly have you take a look here. So these are red blood cells, and when they're in a hypertonic environment, which I'll explain in a second, it means that there's less water outside of the cell than inside of the cell. So you can see water is moving out, okay? This might happen if we have a really, really, really concentrated extracellular fluid here, okay? Lots and lots of solute. That means there's less water, so water's going to move out. Okay, isotonic, iso means equal. So here we have equal movement of water inside and outside of the cell, which means our concentration gradient is the same inside and outside. Sorry, my dog is has severe separation anxiety. So she's sitting up on the bed with me right now. So hi, Addie. And then hypotonic is when we would have a higher concentration gradient or higher concentration of solute inside of the cell, which is drawing water in. Okay, and you can see that those cells are bursting. So again, I'm going to give you more examples and I'm going to write that all out for you in just a second, but I just want you to see what happens to our blood cells. So they can either shrivel or they're nice and happy or they're bursting. The reason I say that is because we're going to take a quick look before we jump in to some more examples at what this looks like in a plant cell. So even if I didn't tell you this was a plant cell, you should be able to tell because we have that giant vacuole in the middle, which is holding water, and we have a cell wall. Okay, so plants do not do well without the right amount of water. Okay, we know this, they shrivel, they wilt, all sorts of things. And so what you can notice here is that plants are actually very capable with being happy in a hypotonic environment all the way to the right because they have that water storage vacuole. They're able to survive with more of an excess of water than our cells are because our cells just burst due to the lack of the cell wall, okay? They're also totally fine in an isotonic state, but they do not do well with hypertonic states. And you can see that plasma membrane is pulling away from the cell wall, and we call that plasmolyzed, which is just a fancy way to say that. So you're asking a great question. So water is still moving with its concentration gradient, okay? It just so happens that where there's a higher concentration of other stuff, there's a lower concentration of water. So hopefully when I pull up my whiteboard in just a second and start writing some things out, you'll understand what I mean a little bit better. But this is kind of why students can sometimes get confused because water is still moving with its own concentration gradient. It just so happens that that's kind of the reverse of the concentration of the other stuff that's around. Okay, so that's the brief answer. I'm going to show you on this like little whiteboard app in a second. And if it still doesn't make sense, re-ask your question and I'll try to do a better job explaining. Okay, so I'm going to show you then what this would actually look like as far as molecules of water and solutes. So I'm going to really quick switch over. Okay. All right, so you should see, whoa, oop, in a second. There we go. This little whiteboard feature. So I'm going to use this to kind of, because I don't know, I need a whiteboard, but this does the trick. Okay, so I'm going to go through all three scenarios with you just to show you, and then we'll talk about some real life examples. As I said, like this comes up all the time in medicine and all sorts of cool things. Okay, so let's start out. Ooh, look at all these pretty colors. Okay, very exciting. All right, so let's start out. Ooh, why? Oh, because I need my pen. Okay, there we go. Sorry, I don't usually use this program. Okay, so let's start out with a hypertonic solution. So a hypertonic solution means two things, okay? First of all, I'm talking about the solution, not the cell. So the first thing that you need to know is that there's more, this is like not calibrated. Okay, more solute outside, yikes, sorry guys, of cell. Maybe I can make my pen point a little smaller. We'll see if that helps. So there's more solute outside of the cell, okay, which means there's more water inside. Okay, so the reason I say that is because solute and water, they both take up space, okay? So if we have more solute somewhere, that means we have less water there. So if we have more solute outside of the cell, it means we have less water outside of the cell. So let me show you a picture. So let's say this is our cell, and these purple guys are our solute. So looking over on the left, this is saying we have more solute outside of the cell. Okay, so let's say the inside of our cell is 15% solute. Okay, and the outside of our cell is 75. That's a crazy big number, but that's okay. We'll use it. 75% solute. Okay, so we have a lot more solute outside than inside. So with those percentages, right, our percentages always add up to 100. So that means the remaining needs to be water, right? So if 15% of our cell is solute, that means the other 85% is water, okay? Then on the outside, the remaining 25% would be water, okay? And this is just a hypothetical example. I just want to show you, I like to use percentages when I first start teaching these concepts, just because it's easier for students to see when I give you the percent. Will you see questions like that all the time? No, but at least you can go through the thought process yourself, okay? And now we can easily see. So I told you guys that water will always move to where there is more solute, but that's also where there's less water, right? So water is still moving with its concentration gradient. It will move outside of the cell here to where there's more solute and therefore less water. Okay, we're going from 85% water to 25% water, high concentration to low concentration. Okay, so what's going to happen to the cell? It's going to shrink. Okay, it's going to shrivel and it could become non-functional. So this is what would happen if you were super dehydrated. If you're really, really dehydrated, the amount of solute in your bloodstream, the sodium and potassium in your blood, that number doesn't really change, but the amount of water does. So it gets more concentrated, okay? Higher percentage is solute. So water can leave your cells and they can start to shrivel up. This also obviously happens with plants, and I'm sure you've seen that or can think of an example. So my fun little mnemonic to remember hypertonic is that when you're hyper, you run outside, right? When you're hyper, you run outside, okay? Water is moving outside to where there is more solute, okay? So that's how I like to remember that. And then again, water's still moving with its concentration gradient from high to low. Some students like to remember that. Some students like to remember that water moves to where there's more solute, It really depends on the student, but those are two examples for you of how water would move in a hypertonic solution. Any questions on that one before we do iso and hypo? Okay. All right, so then can I make this guy bigger? Still figuring this out. I feel like there should be an easier way to erase all of this, right? But the good news is iso is really easy because we know iso is equal, okay? The only kind of important thing to remember that sometimes students forget is water still moves. It just moves equally in and out in an isotonic solution, okay? All right, so flip back here. Isotonic. Okay, in an isotonic, we have equal amounts of water and solute in and out. Okay, equal amounts of water and solute in and out. So what would that look like? So maybe here's our cell, and maybe our cell is 15% solute again, and 85% water. But this time the outside solution that we're in is also 15% solute and 85% water. So there's no concentration gradient here, right? Because we're equal. So water will just move in and out depending, it might shift a little bit, or molecules also just move around, right? They're not stationary. So a couple might bounce in, a couple might bounce out, but there's no we say there's no net movement of water, okay? There's no like grand motion of water moving one way or the other. It's moving equally in and out. So that's the easy one. A really good example of an important isotonic solution. If you've ever had to get an IV of fluids for whatever reason, or you know about IVs in the hospital, and the solution that you get when you get an IV is not pure water. If it was pure water, we'd be in a hypotonic state, which I'll talk about in a second. Instead, it's 0.9% solute. They use sodium chloride, but it's 0.9% sodium chloride. So it's salt water. And that is so that our cells are in an isotonic state, right? Because there's stuff inside of our cells. So the fluid that we're adding should match that. So this would be kind of an example of that, except it's 0.9% instead of 15%. Okay, so there's a real life example for you of when an isotonic solution would be very important. Okay, so then the last one is hypotonic. And my creative way to remember that is hypo-hippo, because these cells get bigger, just like hippos are big. Okay, so in a hypotonic, this is just going to be the opposite of our hypertonic environment. So we have more solute inside, okay, more, gosh, this is like so delayed, water outside. Hopefully you can at least still attempt to see what I'm saying there. I don't know why it's so bad at writing. So more solute inside, more water outside. So what does that look like? Okay, so again, this is like the opposite of our original scenario. So let's say we have 25% solute in this cell and 75% water. Okay, then outside we have 75. Oops. No. Outside, let's say we have 15% solute and 85% water, okay? So I'm writing the percentages of water for you. I will tell you that usually your teacher or the college board, they will not include that. They might just say that there's a higher concentration of solute inside of the cell than in the environment, okay? So you would need to be able to say one of two things in your brain, okay? Water is going to move to where there's a higher concentration of solute, or you can draw yourself a picture and say, okay, if there's more solute inside of the cell, that means there's less water in that cell, and therefore water will move into that cell. Okay, so whatever way you need to think about it, there's a couple different ways to think about it. But water's following its concentration gradient, and that leads it to where there's a higher concentration of solute inside of the cell. So again, I think hippo, because hippos are like big and swollen and lazy. And if you're a hypo, lazy hippo, you're going to go inside and lay on the couch all day. So water moves inside when it's hypotonic. Water moves outside when it's hypertonic, because when you're hyper, you run outside, okay? And water moves both in and out equally when it's isotonic. Any questions about that? I have some practice questions for you, so don't worry. This is not the last. We will talk about it. Feel okay? Okay, so I'll probably pop back to the whiteboard at some point, but let's really quick just look at a few examples just to make it sticky in our brains. Okay. And I picked 3 really important examples. So on the left-hand side, you see sodium chloride 0.9% injection. So that's an actual IV bag that they would use for individuals as long as they're just trying to regularly hydrate them. Because again, if we give them pure water, your cells are all at risk of exploding, right? Because there's a higher concentration of water. It's 100% water, and your cells are not 100% water. So water's going to come rushing into your cells. They can burst. This middle example here is where water regulation comes into play with plants. So obviously I already showed you that plants are actually really okay with being in a hypotonic environment because of their cell wall. There's actually this really cool situation. So all over a plant's leaves are these little holes called stoma. They're little openings, and they're for gas exchange, right? We need to bring in CO2 and release oxygen because they're the opposite of us, okay? And they need to also release water vapor. So they do a lot of exchanging of gases with the environment. So those stoma will be nice and open when there's lots of water present in the cell. And you can see that the water in blue there is actually expanding these guard cells, is what they're called. It is actually forming the opening of the stoma so that the plant can release gases and also some of that water vapor. When water resources are low, okay, you'll notice that these cells kind of close up. So water leaves because we don't have enough water. So you can see here, we have all of this solute on the outside of the cell instead of inside of the cell. So water is leaving, okay, and that actually shrinks up our guard cells and closes the stoma. Why would we want to close the stoma when water levels are low, which is what we're seeing on the right-hand side? Because we lose a lot of water vapor through the stoma. So when water is low, that closes so that we don't continue to lose more water. Plants are very smart, okay? So that's another important example with plants. And then on the right, we have a lovely paramecium, and I can write that in here in case you want to do some research on your own. Maybe you've seen one under a microscope before. They're really cute single-celled organisms, single-celled eukaryotic organisms. But the cool thing that they have is this contractile vacuole, okay? And that actually allows them to pump out water. So let's say they swim into some fresh water and they're used to being in salt water or something like that, okay? That's a hypotonic environment. There's more water outside than inside. So water's gonna come rushing into this little paramecium. He's now at risk of exploding. That would be tragic. So his contractile vacuole can actually pump out that extra water so that he does not explode. So it's kind of cool. You can think about like it would not be very active if he swam into like a really salty ocean, right? Because water's not going to be moving in so much. It actually might be leaving depending on the concentration gradient, but it's just kind of a cool adaptation. And that, for whatever reason, seems to show up on the AP exam a lot, the lovely paramecium and its contractile vacuole. So now you know a little bit more, but these are all real life examples that happen all the time with the movement of water between concentration gradients. So you can just kind of keep those in mind. Sometimes they show up. Okay, so here's some practice. So I have 8 practice questions for you guys to start, where I give you a statement and you need to tell me iso, hypo, or hypertonic solution. So I didn't set this up super well. So why don't I give you like 2 minutes to read through and answer the 8 questions, and then we'll go through the answers. Okay, so don't type anything in quite yet, because then sometimes people see and change their mind. So just take a minute or two to read the questions yourself and write down your answers, and then we'll go through them. Take 30 more seconds. All right, so first question, if you didn't get through all of them, you can also think of them as we're going through. First question, the concentration of the solute in the solution is lower than the concentration of solute inside of the cell. So 2 ways to think about this: water moves towards more solute, or where there's less water. So yes, Elena, you are 100% correct. That is a hypotonic solution, okay? Water moves inside of the cell, that cell will get larger. 2, when a cell is placed in the solution, water will enter the cell by osmosis, causing it to swell. Hypo again. Good. Hypo, hippo. Remember, water moves inside. That cell gets bigger. 3, the concentration of the solute in the solution is the same as the concentration inside the cell. Iso. Awesome. Good job, ladies. 4, the concentration of the solute in the solution is higher than the concentration inside the cell. Hyper, perfect. 5, when the solution is injected into the body, no cell disruption occurs because no net osmosis occurs. Iso, awesome. 6, putting a plant in this solution will result in water loss and cause the plant to wilt. Hyper, good catch there. So that's going to be hypertonic, because water is leaving. Remember, when you're hyper, you run outside. So if you water your plants with salt water, my students have done this as an experiment, water will actually leave and that plant will shrivel, okay? So hyper, because water's leaving. 7, the cell will shrivel when placed in this type of solution. Any answers? Hyper, good. Awesome. And then 8, when a cell is placed in this type of solution, there's an equal amount of water moving in and out. Iso, perfect. Any questions about any of those? I have 2 more practice problems for you. Okay. I mean, you guys crushed it, so nice job. All right, so I have 2 different examples here for you. On the left, I have a beaker with water. Concentrations given, so you need to fill in the blanks, right? So if that cell is 85% water on the left, that means it's 15% solute, okay? And then 95%, and then obviously 5% solute. Same with on the right. So I want you to just tell me, so take a second and then tell me if water would move in or out. So if it's, you can just tell me if it's hyper, iso, or hypotonic solution. All right, so on the left-hand side, what do we have? Okay, so Elena has weighed in. Any other opinions out there before I give away our answers? Yeah, so our first example is actually hypotonic, okay? Remember that water always moves from high to low, so water is going to move from 95% in to 85%. That's also where there's more solute. There's 15% solute in there and 5% salt out. So that's hypotonic, water's going in, okay? And then the second one is also hypotonic, okay? Water will be moving in as well to where there's more solute and less water. So that would be 90% water outside and 82% inside. So water would move into that cell. Any questions about either of those examples? Okay, so usually so these are my own practice problems. Usually I try to incorporate more problems from College Board, especially free responses, but this just doesn't come up a whole lot, especially not on free responses. So it might be embedded in a question in some fashion, maybe in a multiple choice question. But for the most part, this kind of high level overview is enough to get you through. Okay. Any questions? Is there a stream on water potential? That's a great question. So we don't have one scheduled, and I'll tell you why. So water potential usually does not show up on the AP exam at all. So it's not, a great use of time. If it does show up, it used to show up in these questions called grid-in questions, which were math-based at the end of the AP exam. And that section, as of this year, no longer exists. So I've never seen a water potential question show up in a multiple choice question. And it's kind of a confusing topic. And we've just never seen it unless it's in a grid-in, which is like once every 3 or 4 years, which again, they no longer exist. So we don't have one scheduled. If we have extra time or an extra slot, unfortunately, I would have added it tonight, but I came in last minute. So I didn't have enough time to get like a whole lesson on that. But if we have some extra time, I can even try to embed it into our Unit 2 review next Sunday and just talk about it very briefly, because it's really straightforward if you look at the equations that are given, but it just sometimes seems to overly confuse students. And if I had to bet, I would bet a lot of money that it will not be on your AP exam. So that's kind of the rationale there, but I can try to incorporate it a little bit into our review next week now that I know that that is an interest for people. Any other questions? Feeling okay out there? Okay. So I know this was kind of quick. Again, I was pulled in last minute, so this is, I promise you, what you need to know about this kind of stuff, but it also just builds a lot upon like facilitated diffusion and transport types and all sorts of other things that we've talked about over the past week or two. So this is really just like the final like technical vocabulary kind of situation. But if you guys don't have any other questions, if you have some, you can type them into the stream and I'll still answer them. Otherwise, I hope you have a great rest of your Sunday night. Get some good rest and have a great week of school. Bye, guys.